Have you ever wondered how we can actually see the microscopic instruction manuals hidden deep inside our cells? Like, how do scientists zoom past the cell wall, right past the nucleus, and directly highlight the exact sequences of our DNA? Welcome to this explainer. Today, we're going to act as molecular detectives. We're exploring a seriously fascinating biological technique that literally lets us light up the unseen, giving us a rapid, vibrant look right into our genetic code. We're decoding DNA with a process known as FISH. In this explainer, we're going to unpack exactly what this technique is, how it uses these incredibly vibrant colors to tag specific genetic material, and why the exact phase of a cell's life, well, it completely changes what we end up seeing through the microscope. We've got some amazing concepts to cover today, from tightly coiled chromosomes to glowing molecular tracking devices. Section 1. Lighting up our DNA. Okay, before we can track down a specific genetic sequence, we need a background canvas, right? To create that canvas, scientists use a dye called DAPI. Think of DAPI as a universal highlighter for genetic material. It stains all of the DNA in the cell this deep, brilliant blue. Now, depending on what phase the cell is currently in, this blue signal is going to look really different. Sometimes it gives us a wide, diffused background signal, and other times it highlights tightly condensed structures. But honestly, regardless of the phase, this blue DAPI stain is the absolutely crucial first step that makes our entire genetic landscape visible.
Once our blue background is set, we bring in the star of the show, fluorescence in situ hybridization, or thankfully, FISH for short. FISH uses glowing, fluorescent probes that act exactly like targeted molecular tracking devices. You basically unleash these probes into the cell, and they hunt down and bind to highly specific genetic sequences. So picture this. Against that blue DAPI background, these probes light up like neon signs, pinpointing exactly where your target DNA is hiding out.
Section 2. Metaphase, the condensed view. Metaphase creates a really distinct crime scene for our molecular detectives. When we perform FISH on a cell in metaphase, we're catching the cell right when its DNA is tightly condensed. It's kind of like a traveler who has intensely packed all their clothes into tightly zipped suitcases right before a huge trip. Because of this really tight packing, the blue DAPI stain lets us clearly see individual chromosomes. Plus, in metaphase, DNA replication has already happened. The suitcase has essentially been duplicated, meaning what we call sister chromatids are now present.
2. I really want to emphasize that number. Because the DNA has already replicated, in metaphase we get 2 signals per chromosome. When our glowing FISH probe actually finds its targeted region, it binds to both sister chromatids. So for literally every chromosome you target in metaphase, you're going to see a distinct pair of glowing spots.
But, nature is incredibly dynamic, and timing is absolutely everything. Depending on the exact fraction of a second during mitosis when the cell is fixed or frozen in time for the test, those metaphase chromosomes might look slightly different. Sometimes the 2 sister chromatids can be really easily distinguished from one another. Other times, they're bound so tightly together that they actually just appear to be a single thick chromosome arm. It totally depends on exactly when the molecular detective snaps the photo. Section 3. Interphase, the diffuse nucleus. Now, if metaphase is the cell all intensely packed travel, interphase is the cell just relaxing at home. It's the growth phase. The DNA isn't packed away at all. It's totally unpacked and actively being used. The genetic material is entirely decondensed. Because of this, when we use our blue DAPI stain during interphase, we actually don't see distinct separate chromosome structures. Instead, the decondensed DNA just kind of fills the entire nucleus, creating this wide, soft, and diffused blue signal across the whole area. 1. And here is a really stark contrast to what we just talked about. Remember how metaphase gave us 2 signals per chromosome because the DNA had replicated? Well, in interphase, the DNA has not yet replicated. So, when our glowing FISH probe hunts down its sequence, each targeted chromosome is going to be represented by just a single, lonely fluorescent spot floating against that diffuse blue background. So let's compare this visual divide side by side. On the left side, we have metaphase FISH, tightly condensed individual chromosomes showing the results of replicated DNA with those sister chromatids. Then, on the right side, we have interphase FISH, decondensed, unreplicated DNA forming a diffuse background with just single probe spots. The state of the cell completely dictates the canvas our molecular detectives are working on. Section 4. Reading multiple color probes. Now, you might be wondering, why settle for tracking just one sequence when we can track several? To do this, we use a process called multiplexing. Multiplexing allows us to send multiple fish probes into the cell at the exact same time. The only catch? They absolutely must have different fluorescent markers. By using distinct colors, we can hunt for totally different genetic sequences simultaneously, all without getting our signals crossed. And this brilliantly illustrates exactly how we map multiple locations in this example. We unleash two completely different probes into our DAPI-stained cell. We have a green probe specifically engineered to bind to a sequence on chromosome 9, and a red probe designed to target a sequence on chromosome 22. When we look through the microscope, we can instantly differentiate the location of chromosome 9 from chromosome 22 just by looking for those green and red glowing beacons. It's that straightforward. However, as with anything in science, there are always trade-offs. Metaphase fish, because the chromosomes are so tightly condensed and distinct, offers incredibly precise localization. I mean, you can see exactly which specific region of a chromosome your green or red probe is binding to. But, and this is a pretty big but, it demands perfect timing. It requires the cells to be in that very specific, very brief metaphase phase of the cell cycle to actually work. Interphase fish doesn't require that perfect timing, but you do lose that highly specific chromosome localization.
Section 5. A rapid diagnostic test.
So, what does this all mean? Why does all this microscopic tagging actually matter in the real world? Well, in a clinical setting, time is almost always of the essence. Both metaphase and interphase fish are incredibly valuable because they act as rapid tests. Unlike some of the older methods that might literally take weeks to grow and analyze, the fish technique can be completed in a very short time frame. This gives doctors and scientists a super fast, frontline glimpse into a patient's genetic makeup. But we have to remember that fish is really just step one in the diagnostic pipeline. While fish gives us that incredible, fast-acting, glowing snapshot, it's still considered a rapid screening tool. Any findings that we discover using fish ultimately need to be confirmed by step 2, a definitive diagnosis using more comprehensive methods, like a full karyotype or a microarray analysis. Basically, fish tells us exactly where to look, but we still absolutely need those broader tools to confirm the whole story. And that brings us to the edge of our molecular map today. We've seen exactly how fluorescent probes act as brilliant tracking devices, lighting up our genetic code against a blue DAPI background, whether that's in the tightly packed luggage of metaphase or the soft, diffuse expanse of interphase. But it leaves us with a really exciting frontier. While fish gives us a rapid, targeted glimpse into our cells, what other profound secrets does a full, definitive karyotype reveal about who we actually are? Keep wondering, keep learning, and I'll catch you on the next explainer.